Hello everyone, welcome back to my channel Science with Sangati. So I hope all of you are doing so well in your life. So today we are going to discuss about the topic cell, the unit of life. Okay, all of you can take out your notebook so that you can note down the important points, and let's start today's session. Today we are going to cover the prokaryotic cells and the difference between prokaryotic and eukaryotic cells. So let's get started. So cell, what do you understand by the term cell? So you might have learned about this thing in your junior grades also, right? Lower grades. So what is a cell? Cell is the unit of the life as the chapter's name is saying itself. So basically how the cell is the unit of a life. So you know that a building is made up of so many bricks, right? So maybe 10,000s of bricks are required to make a building. So similarly, the living organisms are made up of so many cells. That's why it is called as the building block. Okay. So like our body, any other organism's body, dog's body, cow's body, every organism's bodies are made up of cells only. So animals has a different types of cells and plant has a different types of cells which we call as a plant cell. In the animal cell also we can see that there are so many different types of cells are there like RBC, WBC, right? So there are so many different types of cells are present. We are going to discuss about all of these things. Okay. So cell is making all of the organism. It is thus basic structural and functional unit of life. Okay, because in our body, whatever the functions we are doing, like digestion, respiration, all of these functions are actually performed by the cells only, right? You know, everything is happening inside the cells. There are different types of cells in our body, correct? But do you know what the cells are made up of? What? We are made up of cells, but the cells are made up of what? The cells are made up of the basic structure, basic unit, that is the atom. Okay, because everything in our world is made up of atoms only, right? If you want to know more about the atoms, you can click on here in the I button over here. Okay, so you will get to know more about the atoms, like who discovered the atoms, how how many subatomic particles are there. Okay, you can click on this video. Now atoms made the molecules. Okay, so many different types of atoms combines together to form the molecule, and from the molecule we get the macromolecule. From macromolecules combine together to form the organelles, and organelles combine together to form a cell. Similar types of cells, so performing same functions, they combine to form the tissues combine to form the organs, like we have liver, stomach, intestine, so many organs. And organs combine to form the organ system, like we have digestive system. So liver, stomach, intestine, they are the part of digestive system. Okay, we have respiratory system, right, which includes the lungs. Then we have other systems, excretory systems. So there are different types of organ systems, and all of these types of organ systems combines to form a complete organism. Clear? So this is how everything is, builded. So that's why cell is the building block of life, but atom is the smallest unit of every matter. Okay, so basically cell are also made up of atoms only, because we are also the matter. Now, can you tell me who discovered the cell? The first cell was discovered by Robert Hooke. He discovered the dead cell in the year 1665. Okay, and which kind of cell he discovered? He discovered a cork cell in the plant. Okay, then the living cell was discovered by A.V. Leeuwenhoek in the year 1674. That was the free living cell. He found Spirogyra in the pond water. Then Robert Brown discovered nucleus inside the cell in the year 1831. Please don't get confused between Robert Hooke and Robert Brown. Both of them are like different scientists. Please remember these terms. Purkinje coined the term protoplasm for the fluid substance like the cytoplasm and the nucleus, both together called as protoplasm. Then comes the very important, that's the cell theory. Schleiden and Schwann proposed the cell theory. Schwann was zoologist, so he said that all the animals are made up of cells. He was zoologist, so he was like studying on all the animals. He said that all the animals are made up of cell. Schleiden was botanist. Schwann was zoologist. Schleiden was the botanist. Both were German, and he said that all the plants are made up of cell. So both of them, when they come together, they said that okay, plants and animals, that means all the organisms, all the living organisms are made up of cells. Okay, so that was the first point of cell theory. Second point, both of them together said that all the cells, all the the cells are basically the structural and functional unit of life. So cell is providing us the structure, and all the functions are also performed by the cell. So that's the second point of the cell theory, and both of them proposed these 2 points together. Okay. Then Rudolf Virchow in the year 1855, he came and he did some he observed the cell division process under the microscope, and he said that okay, all of these cells are arising from a pre-existing cells. So that means there was one cell already, and that cell divided into two different cells, right? So these two cells are arising from a pre-existing cells. Okay, so Rudolf Virchow expanded the cell theory and added this point. Okay, so these are all about the discoveries of the cell and its components. Okay, now let's move on to the different types of cells. So on the on a basis of the size, okay, we have divided the cells into two categories, basically the prokaryote and the eukaryote. So there are two different types of cells, prokaryotic cell and the eukaryotic cell. Prokaryotic cells are very, very small, and they are kind of primitive cell. Okay, just remember that primitive, very primitive. You know, the earliest cell which develops on the earth. Eukaryotic cells are a kind of new cells. Okay, now in the prokaryotic cells, basically you can see the structure over here. So prokaryotic cell is having a capsule, cell wall, cell membrane. So there are three outer coverings: capsule, cell wall, cell membrane. In the eukaryotic cell, and especially in the case of plant cell, there are two outer coverings, cell wall and cell membrane. In the animal eukaryotic cell, only cell membrane is present. Okay. So prokaryotic cells are having three outer coverings. They do not have membrane bound organelles. Even the nucleus is also do not have any nuclear membrane. So that's why there is no intact nucleus. So the genetic material is just spread in the cytoplasm. Okay, and that type of genetic material is called as nucleoid. It's just spread. Now, prokaryotic cell is having another type of DNA that is called as the plasmid DNA. And this type of DNA is a self-replicating DNA. It's a circular DNA, plasmid DNA. Prokaryotic cells have ribosomes on it, okay, which is not membrane bound. Even the eukaryotic cells ribosomes are not membrane bound. Okay. Now, they don't have so much of cell organelles, which you can see eukaryotic cells has so much of cell organelles like mitochondria, centrioles, Golgi apparatus. Prokaryotic cell do not have that much of cell organelles. They have the pili and the flagella. Okay, so we are going to discuss about the structure of the prokaryotic cells in details. Just wait for a few minutes and watch the video till then.
Cells are the smallest living units of an organism. All cells have three things in common, no matter what type of cell they are. All cells have a cell membrane, which separates the inside of the cell from its environment, cytoplasm, which is a jelly-like fluid, and DNA, which is the cell's genetic material. There are two broad categories of cells. The first category is eukaryotic cells. They have organelles, which include the nucleus and other special parts. Eukaryotic cells are more advanced, complex cells, such as those found in plants and animals. The second category is prokaryotic cells. They don't have a nucleus or membrane-enclosed organelles. They do have genetic material, but it's not contained within a nucleus. Prokaryotic cells are always one-celled or unicellular organisms, such as bacteria.
Alright, I hope you have enjoyed the video. So let's see few differences between the prokaryotic cells and the eukaryotic cells. So first of all, as I told you that prokaryotic cells are very small size, like 0.125 micrometer in size, whereas the eukaryotic cells are larger, 10 to 100 micrometer in size. Okay. Prokaryotic cells are like unicellular only. For example, the bacteria, archaea, they all are unicellular cells. But in the eukaryotic cell, we have both multicellular as well as unicellular. For example, the protistas, they are eukaryotic cell, but they are unicellular. Okay, but the plants, fungi, animalia, all of they are multicellular. Okay, but yeah, yeast is a fungus which is unicellular. Okay, so there are both types of cells are present in eukaryotes. Prokaryotic cell, nucleus is absent. So what is present? What is present? Tell me the answer. We just discussed. Nucleoid is present. Okay, so the proper nucleus is absent, but the genetic material is just present over there in the cytoplasm, which is called as nucleoid. Whereas in the eukaryotic cell, nucleus is present, proper nucleus. Prokaryotic cell is having a circular DNA, and the name of the circular DNA is the plasmid DNA. But the eukaryotic cell has a linear DNA, which is the double helical structure DNA. Prokaryotic cells, only one chromosome is present. One single chromosome is present. So that's why it is haploid. Eukaryotic cell is diploid. There are so many chromosomes are present. It depends on the species to species. So Homo sapiens has 23 pairs of chromosomes. So it is always in pairs. So that's why it is called as diploid. Prokaryotic cells lack membrane-bound organelles, but eukaryotic cells has membrane-bound organelles. Okay. Prokaryotic cell can also reproduce like asexually and sexually both, but eukaryotic cells mostly reproduce sexually, but there are asexual reproduction also is there. Okay, but mostly they reproduce sexually. In the prokaryotic cell like bacteria, the cell division occurs by binary fission process. In the eukaryotic cell, the cell division is by mitosis process. Okay, mitosis and meiosis, both the types of process are there. Examples of prokaryotes are bacteria and archaea. Examples of eukaryotes are plants, fungus, animals. Now there are some similarities. So basically, both the types of cells are having cell membrane, cytoplasm, genetic material, and ribosomes. So these four are present in both the types of cells. That's why we can also say that these are the basic functional unit of the cell. Because from the prokaryotic cell to now the modern cell, like all these cells are having these structures in common. So that's why we can call it as a basic functional unit of the cell. Okay, I hope this is clear to all of you. So these are all about the differences between prokaryotes and the eukaryotes. If you want to take some more notes, you can pause the video over here and you can write down the differences from this chart. Okay. Now, so here you can see what is the last, what was our ancestor basically. Here you can see this tree. What do you understand by this tree? Does that means that we and the bacteria have something in common? It's funny. It sounds funny like bacteria is something having common with to us, you know. But yeah, there is something common. 1% of the DNA is common. Yeah. So this traces back to the LUCA. What is LUCA? The last universal common ancestor. Okay, that is called as the LUCA. So we don't have any fossil of LUCA, particularly because of course it was billions of years ago. But because we saw after doing so much of research on the DNA of every organisms, we saw that there are some common factor is there. So that means we all had same origin. Okay, we all had a same common ancestor and we named it as a LUCA, last universal common ancestor. Now from the LUCA, you can see there are three domains. One domain is the bacteria, another is the second is the archaea, then there is the eukarya. Now in some books like NCERT, bacteria and archaea, they are considered to be, together only. But according to the modern classification, so they falls in two domains, like basically they are different. Okay, they are not one. Bacteria and archaea are not one, they are different. Okay, so we consider it as a second domain and the third domain is eukarya. All right, so bacteria and archaea, both of them are prokaryotes only. Both of them are prokaryotes, but eukarya is eukaryotes. Okay, they are the eukaryotic organisms. Now in the bacteria you can see like there are several examples: gram positive, gram negative cyanobacteria. In the archaea there are like methanobacterium, thermococcus, thermoacidophilus. Okay, so these are the archaea. Now yeah, there is some difference. We are going to discuss about the difference between bacteria and archaea also. A little bit minor difference is there present in the cell wall and the cell membrane. That's why we have considered the separate domain for the archaea, because the structure is basically different. First, let's talk about the bacteria. Then we are going to discuss about the archaea. Both of them are prokaryotes. Remember that. Okay. So bacteria is the prokaryote, it's of course unicellular because all the prokaryotes are unicellular and they are found almost everywhere, like everywhere in your body and on your skin, on your table, everywhere it is there. Okay, you cannot see that. So we have the like fossil evidence of the last bacteria is around 4.3 billion years ago. The oldest fossil of bacteria which we have got. Okay. So just imagine LUCA was before 4.3 billion years ago, right? So now they mainly reproduce by fission, spore formation under the unfavorable condition. But on the favorable condition, binary fission is suitable for the bacteria. Okay. They can also do the sexual reproduction by transferring the DNA, okay, from one bacterium cell to another bacterium cell. All right. Mycoplasma lacks the cell wall and they are the smallest cell, which can survive without oxygen. So just remember, mycoplasma is the smallest cell. What is the largest cell then? Ostrich egg, right? Ostrich egg is the largest cell. What is the longest cell? Nerve cell is the longest cell. Okay, especially human nerve cell is 1 meter long And if I talk about blue whale's nerve, it's 25-meter long. So the longest nerve cell is blue whale's nerve. Okay, just remember that. So that's about the like different types of cells according to the size. Okay, largest cell in human being is the ovum, the egg. Okay. Now, there are different types of bacteria. First category is the photosynthetic autotrophs. So here you can see the example of cyanobacteria. Cyanobacteria is nothing but it's the it's cyanobacteria is basically a blue green algae. The common name is algae. It's not a algae. Okay, just remember that it's like bacteria only. So they have this structure. They have this chlorophyll and the carotenoid pigments are present. You can see they have this thylakoid structure over here, all right, which is having this chlorophyll and the carotenoid pigment. Again, they don't have a proper nucleus, so this is the reason that we do not consider this in the algae category. Algae comes under the Thallophyta, right, in the plant kingdom, but we do not consider this because it does not have a proper nucleus, right? It's not eukaryote, it's prokaryote. So that's why we call it as a bacteria, cyanobacteria. So it is having this nucleoid, and the important thing is they have a mucilaginous sheet. You can see there is a mucoid sheet surrounding. All right, it's a slimy sheet surrounding. All right, it is unicellular and filamentous. They are present in colonies. Okay, for example, the examples are Anabina and nostocs. They can also help in the nitrogen fixation process. These are mainly present in the, you can say, the fresh uh, pond waters. All right, like non-polluted pond waters, especially. They are not present in any kind of pollution. All right, so if we see that okay, this water is having cyanobacteria, that means we can consider that okay, this water body do not contains any pollution. All right, now there are next types of bacteria like the chemosynthetic autotrophs. They they require the energy for the breakdown of their food, and they get their energy from various inorganic substances like ammonia, nitrates, nitrites, or the sulfur also sometimes. Okay, those are the chemosynthetic autotrophs. Next comes the heterotrophic bacteria, which also acts as a decomposers. Okay, for example, the nitrogen fixing bacteria are there, the lactobacillus acidophilus are there. They helps in fermentation, and there are some other bacteria. They helps in the antibiotic production. Citrus canker is there, tetanus. Okay, so they can cause the diseases also. So there are different categories of the bacteria. Now let's see the different shapes of the bacteria especially. So shapes, according to the different shapes, again we have different categories. First is the cocci. Cocci means C for cocci, C for circle. All right, means they are spherical in shape as you can see in this diagram. So spherical. Now this spherical shape can be alone, they can be all just connect together to form a elongated spherical structure like this, or they are present in some like colonies. So coccus, wherever you see the term coccus, that means these bacteria are having spherical shape like streptococcus, staphylococcus. Okay, streptococcus pneumoniae causes pneumonia, staphylococcus aureus. So these are the different types of like spherical bacteria. Next comes the rod shape. Rod is called as the bacillus. All right, bacillus or bacillus, whatever you can pronounce, bacillus. So like bacillus anthracis, it causes anthrax. Where? Especially in the cattles. Okay, they cause anthrax disease. So then salmonella, this is also a rod-shaped bacteria. Clostridium botulinum. Okay, do you know what is Clostridium botulinum? This is the bacteria which is basically used in the buttocks. If you know about Botox, it's in modern days, every celebrities are doing Botox to reduce their wrinkles. So basically, Clostridium botulinum is a bacteria. This releases a toxin which is called as botulism. Okay, this releases the toxin which helps to paralyze. Of course, it is not good for us, so that's why. So basically, it was first used as a test for the cancer disease, but the doctor said observed that it is causing the paralysm. Okay, the person is getting paralyzed in case of overdose. But now, because we are human beings, we have a great mind, we are using that thing to rectify our muscles. So basically, this toxin, what it does when we inject it in our muscles or in underneath the skin, in the Botox treatment, it paralyzes the muscles over here. So it do not cause the wrinkles. Okay, it reduces the wrinkles. That's how it is used in the all of these, like these what you can beauty cosmetic surgeries. These are used. Of course, it should be used in a like proper dose, because the overdose can cause paralysm. Okay, paralysis. Next up is the spiral shaped bacteria. These are called as the Vibrio. Okay, the spiral, the Vibrio are the basically comma shaped, spiral shaped are the spirillum. Okay, Vibrio are comma shaped. So, Vibrio cholerae causes cholera. Spiral shaped, the spirillum, like this one. So, they are the spiral shaped bacteria. Heliobacter pylori, it causes the ulcer. So there are different types of bacteria. Again, good bacteria, bad bacteria, both the types of bacteria are present. Okay, let's see the shape of the bacterial cell. So here you can see the bacterial shape structure, and as I told you, it is a prokaryotic cell. So of course it is having three cell walls, three outer covering. It's like extra protection. So the outer covering. So basically, if I draw a diagram over here, these are the outer covering. Three. The outermost covering is made up of glycocalyx. Glycocalyx. Okay. Now this can be two types. One is hard capsule and another is sticky, slimy. Okay. Sticky and slimy. So this can be in two forms, but it is made up of glycocalyx. So here you can see the capsule, that is the outer covering, which is made up of glycocalyx, and capsule is the hard covering. So in different bacteria, different outer coverings are present. In some it could be hard, in some it could be sticky. Okay. The second covering is the cell wall, which is made up of, tell me, peptidoglycan. Okay, the cell wall is made up of peptidoglycan. In the plant, cell wall is made up of cellulose, hemicellulose, pectin, but in the bacteria, cell wall is made up of peptidoglycan. Peptido means amino acids are there. Glycan means sugar are there, sugar compositions are there. Okay, so cell wall is made up of peptidoglycan. The third layer is the plasma membrane. Now, the plasma membrane is like, same only. We also have phospholipid bilayer, so they also have a phospholipid bilayer. But here you can see there are some infoldings in the plasma membrane, and these infoldings are called as mesosome. Okay, so these infoldings are called as mesosomes. Basically, they are increasing the surface area of the plasma membrane. Okay, now why they are increasing the surface area? Why there is need to increase the surface area? Because in animal cell or plant cell, their plasma membrane is like regular only, right? There are no protrusion, like there is nothing called as mesosome. So basically, see, in the plant cell, animal cell, we have a lot of cell organelles, right, to do different functions. But the prokaryotic cell are like, they don't have anything, right? They don't have that much cell organelles. So that unicellular organism has to do every functions, right? So that's why they need to increase their surface area of the plasma membrane so that the mesosome can perform different functions. Okay, so now what are the functions of mesosome? Let's see this over here. Mesosomes are typical enfoldings in the cell membrane that are found in the like prokaryotic cell. They are the extensions. They are like a protrusion, protrusions or extensions you can see in the form of vesicles. Vesicles are circular. Tubules are like tube-like elongated, and lamellae are like this. Okay, in any form they can be. So these are the mesosomes. They are doing the functions like they helps in the cell wall formation, cell division, DNA replication, oxidative phosphorylation, whatever the functions are there in the cell, mesosomes are helping there because they don't have any other cell organelles to do this, right? So mesosomes is doing all of these functions. I hope this is clear to you. Now, next structure we can see that here the nucleoid is present, the genetic material. Ribosomes are present. So prokaryotic cell has 70S ribosomes. Okay, 70S ribosomes. Now what is the 70S ribosomes called? Let's see this. So here is the 70S ribosomes. S means the sedimentation coefficient. Sedimentation coefficient. And what do you understand by sedimentation coefficient now? So suppose you take the ribosomes and you are, you put it under a centrifugation machine and you are rotating it at a very high RPM. It is centrifuging. So what will happen? The heavier particles, more denser particles will settle down at the bottom, and the less denser particle will be on the up, right? So the particles will settle down, so sediment down. So according to that, we have decided the sedimentation coefficient. So 70S ribosome is made up of 50 and 30. 50s and 30s. So the 50s are like bigger size. They are more heavier, more denser. They will settle down at the bottom. And the 30s is like smaller size. They will at the top. Okay. So that according to that, we have decided, according to the density, we have decided the sedimentation coefficient. Now S, this sedimentation of this ribosome, it was discovered by Swedberg. Swedberg. So that's why this is also called as Swedberg unit. Okay, the S is also called as the Swedberg unit. Now you can see that in the prokaryotes, there are two subunits. Larger one is 50S, the smaller one is 30S. So 50 plus 30 isn't should be 80, but it's 70. So that means these are not additive. You cannot just add it, you cannot apply maths over here. Okay, so basically these two types of units, subunits, they are overlapping like this. That's why it takes the shape of 70s together. Okay, so that's about the prokaryotic ribosome. And as you know that ribosomes are mainly helpful in the protein synthesis process. One more thing I would like to add over here, that this 70S ribosome is also present in mitochondria and chloroplast of the eukaryotic cells. Okay, so that's about the prokaryotic ribosome. Now, next structure, what you can see over here, you can see some inclusions bodies are there and some plasmid DNA is there, one circular DNA, plasmid DNA, which is a self-replicating DNA. It also helps to carry, the uh, uh, it also helps to carry, to transfer to any other bacteria. Okay. So now next you can see the pilus. Pili structure are there which is helpful for the attachment to where, suppose the bacteria wants to sit somewhere, or bacteria wants to attach to any host body, okay, or any substratum. So pilus are going to helpful in the attachment process. Now this flagella, okay, now there are two different flagella. So eukaryotic cell flagella structures are different. Prokaryotic cell flagella structures are different. Okay, eukaryotic cell flagella I'm going to describe you when I'm going to teach you about the eukaryotic cell. Today we will be just discussing about the prokaryotic cell flagella. So here the flagella is having this basal body, okay, basal body, and this is the filament. So basal body and the filament. So in the basal body here you can see there is a hook-like structure. Hook is there, okay, which gets attached. So whenever the bacteria is moving, this flagella, this filament moves, okay, and this filament and this complete structure is attached to the bacteria by this hook, basal body. All right. So these are helpful in the movement. All right. Okay. So this is all about the structure of the bacterial cell. Now let's see this video that how a bacteria is moving and it is getting attached. See how it is moving? It is having this flagella, long tail at the back, and the pili are there, and it's the host body, and it is, it's moving, and somewhere it can get attached. These pili are like basically, spikes like structure. So they are helping the bacteria to attach to any surface. Now, prokaryotes do not have cilia. These pili may looks like cilia, because in the eukaryotic cells also cilia looks like this, appears like this, but it's not cilia. Prokaryotic cells do not have any cilia. These are the pili only, or pilus. I hope this is clear to all of you. Now, let's move on to the different categories of bacteria according to the staining. That is the gram-positive and gram-negative bacteria. So under the microscope, gram-positive looks violet stain, and gram-negative is pinkish in color. So basically, what is this stain? Gram stain. So Hans Christian Gram discovered this technique, okay, and he discovered a stain that is called as the Gram stain. And when we are doing the experiment and staining those bacteria, some of the bacteria can absorb that stain, and some of the bacteria do not absorb that stain. So which bacteria which do not absorb the stain, they are simply called as gram-negative bacteria. These gram stains are having violet color. Okay. So basically, how the gram-positive bacteria is going to um, uh, uh, get stained. Okay. So these bacteria are having a very thick layer of peptidoglycan in their cell wall. So as I told you that bacterial cells are made up of peptidoglycan, right? So cell wall is made up of peptidoglycan, but there are some bacteria which are having thicker cell wall, and there are some bacteria which are having thinner cell wall. Okay, so the gram positive bacteria has a thicker peptidoglycan cell wall, and they also have teichoic acid, okay, in their cell wall. Teichoic acid. So, and they also lack the lipopolysaccharide layer. So when we are staining, they can absorb the stain because they have a very thick cell wall. Teichoic acid is there. So they retain the stain, basically not absorb, they retain the violet color. And when we observe it under the microscope, they appear to be violet color. Okay. For example, Staphylococcus, Streptococcus, Clostridium, all of these are gram-positive bacteria. Next is the gram-negative bacteria. They have a lipopolysaccharide layer on the outer membrane, and they do not have teichoic acid. Okay, the peptidoglycan layer is also very thin. This is the reason that they cannot retain the stain, and they cannot retain the crystal violet stain, and they can just stain by the safranin, so they appear pink color under the microscope, like E. coli, Pseudomonas, cyanobacteria are there. So here you can see the difference between the gram positive and gram negative bacteria. Cell wall is single layer, smooth and very thick. Okay, in the gram positive. In the gram negative there are double layers, but very thin. Thickness is very less, 8 to 10 nanometers only. In the gram positive they have a thick layer of peptidoglycan, multi-layered, but here it is thin layer. There, in the gram positive teichoic acid is present, in the gram negative teichoic acid is absent. In the gram positive lipopolysaccharide is absent, but in the gram negative lipopolysaccharide is present. Outer membrane in the gram positive is not present, but in the gram negative outer membrane is present. Okay, so these are some differences. Here you can see gram positive are very susceptible to the antibiotics, but gram negative are resistant to the antibiotics. Here you can see the structure of the membrane. Here you can see this is the like very thick peptidoglycan layer, and here it's very thin. Okay, and lipopolysaccharides are also there in the gram negative bacteria. This is the reason that gram positive bacteria can retain violet stain. Okay, because they have a very thick peptidoglycan layer having teichoic acid on it. So these are the few differences between gram positive, gram negative bacteria. Now for a better understanding, you can watch this video.
This is important since bacteria often experience variations in osmotic pressure due to the different solutions they encounter, and it is their cell walls which prevent them from shrinking or swelling. As a reminder, osmosis is the process by which solvent molecules pass through a semi-permeable membrane from a less concentrated solution to a more concentrated one, equalizing the concentration on either side of the membrane. Nearly all bacterial cell walls have a peptide polysaccharide layer called peptidoglycan, also known as murane. Peptidoglycan is a polymer made up of sugars and amino acids which forms a kind of mesh. Bacteria can be classified based on their reaction to the gram stain, which identifies them as gram-positive or gram-negative, based on the chemical and physical properties of their cell walls. Gram-positive bacteria have a thick cell wall, which consists of up to around 30 layers of peptidoglycan. This cell wall surrounds a monoderm, which is a single plasma membrane. Gram-negative bacteria have a much thinner cell wall, consisting of a single layer of peptidoglycan. This layer of peptidoglycan is sandwiched between two lipid bilayer membranes called diderms. We can differentiate between gram-positive and gram-negative bacteria by dyeing them with crystal violet and then washing them with a decolorizing solution. Then a counterstain is added, for example, safranin or fuchsine. Gram-positive bacteria will retain the crystal violet dye and remain purple, while the gram-negative bacteria will be stained pink. Note that gram-positive bacteria also pick up the pink color of the counterstain. However, this is not visible when they are dyed with the darker purple color of the crystal violet stain. The reason for these staining differences is due to differences in cell wall structure, which is the chief difference between gram-positive and gram-negative bacteria. The gram stain detects peptidoglycan, and since gram-positive bacteria have a thick multi-layered peptidoglycan layer, they retain the crystal violet dye. Gram-negative bacteria do not retain the dye for two reasons. They have an outer membrane getting in the way of the crystal violet, and they lack peptidoglycan to retain the stain. Although both gram-positive and gram-negative bacteria can be pathogenic, gram-negative bacteria are more resistant to antibodies because of their impenetrable cell wall. Unfortunately, these bacteria also develop resistance more quickly. Not all bacteria can be reliably classified through gram staining. Acid-fast bacteria and gram-variable bacteria, for example, do not respond to gram staining. Acid-fast bacteria are bacteria whose cell walls retain stains particularly well. Although they aren't closely related to gram-positive bacteria, they can appear purple after the gram stain test. Gram-variable bacteria show a mix of pink and purple cells when stained.
All right. So I hope it is clear to you. And that's all about the bacteria. Okay. And that's all about our first domain. Now we are going to discuss about the second domain, that is the archaea. Archaea are the prokaryotes. They are also unicellular only, but they survive in some extreme conditions. Okay. And like harsh environmental condition, like it can be salty, marshy, or hot springs. And according to that, like if there are some archaea which are surviving in salty areas, they are called as halophiles. If they are surviving in marshy areas are having methane gas, so they are called as methanogens. In the hot springs, they are called as thermoacidophiles. So there are different types of archaeas. So initially, it was thought that archaea was also bacteria, but in the year 1977, Carl Woese, he gave them a separate domain, and that was necessary. Why that was necessary? Because the structure of the cell wall and the cell membrane is completely different. So we cannot say that archaea are the bacteria. Archaea are different. Okay, so that's why they are given a separate domain. All right, so let's see this. Let's see what are the difference and the similarities between archaea and bacteria. So here you can see in archaea can survive in the extreme environmental condition, but bacteria is present everywhere. Okay, like they can survive in non-extreme condition as well as some uh, extreme condition also. Archaea lacks the peptidoglycan layer in the cell wall, and this is a major difference, whereas the bacteria has a peptidoglycan layer. Okay, the shapes of the archaea can be spiral, rod, sphere, flat, square, plate. But in bacteria, we just get vibrio rods, filamentous cocci, bacillus, spirillum, but there is no flat or plate shape. Archaea lipids, the cell membrane, lipids are ether linked. Okay, ether linkage are there, but in bacteria there is ester linked. In archaea, examples are like halophiles, thermoacidophiles, methanogens. In bacteria, there are two categories, gram positive and gram negative. Okay, so these are some major differences between the archaea and the bacteria. Here you can see in the diagram like this. So this is the archaea. Here is the ether linkage you can see. Okay, and in the like bacteria and eukarya cell wall, cell membrane, sorry, there is the ester linkage. Okay, because we have the phospholipid bilayer, there is the ester linkages there. So there is a huge difference between the cell wall as well as in the cell membrane. Let's see, observe this, watch this video for a better explanation.
If you mention the name Carl Woese to most biology students, you may draw a blank stare. In reality, modern classification had a paradigm shift and added a third domain thanks to his work. He was the first to show how archaea organisms are not bacteria nor a eukaryote, but a completely different domain of organisms. Let's take a look at how bacteria and archaea are different. One of the first things you will learn about archaea is that many live in extreme environments, like deep hydrothermal vents or in hot springs. Now this is true, but they can also be found living next to bacteria in your gut. Archaea do share many similarities with bacteria, which may cause you to think that they are the same organism. They are single-cell organisms. They do not contain a nucleus nor membrane-bound organelles, and they do reproduce asexually. However, there are some important differences. First, the cell walls of bacteria and archaea are different. Bacteria cells have walls that contain peptidoglycan. Archaea cells do not contain peptidoglycan in their cell wall. Cells have membranes that surround them and allow materials in and out and separate them from their environment. The plasma membrane of archaea use isoprene chains instead of the fatty acid chain which are found in bacteria. The enzymes that read the genetic code in archaea are different than the enzymes that read the genetic code in bacteria. I find this interesting. To date, no archaea are found to cause disease in humans. Some diseases we know that we get are caused by a bacterial infection. As a result of these differences, and several others, modern classifications changed and a third domain was added.
All right, so that's a nice explanation. So here you can see the chain is also different. This is the isoprene chain, okay, and this is the lipid chain basically. So this is also the difference. So now there is a one more interesting thing that archaea do not cause diseases in human beings. So they are not pathogen for us. It's a good thing, right? But bacteria, they cause diseases. There are the bad bacteria which can cause diseases which are harmful for us. Okay. Now let's see the difference and the similarities properly. Archaea, of course, they are give separate domain, but in some books, okay, they are given in a along with the bacteria only. So somewhere you can see archaebacteria is written over there, but you can just write over here the archaea, okay. And the bacteria are called as eubacteria because they are the true bacteria, okay. Archaebacteria is the archaea group, and eubacteria are the normal bacteria group, true bacteria. So similarities are both of them are prokaryote, both of them do not have nucleus, proper nucleus, so nucleoid is present, both of them are unicellular, ribosomes are there, no membrane bound organelles are there. These are the similarities. And differences we have already seen. Again, see this peptidoglycan cell wall is present in eubacteria, but it is absent in archaea. Okay, and they cannot survive in extreme conditions, not that much extreme conditions, but archaea can survive in extreme conditions. All right, the cell membrane is also different. Here the cell membrane is made up of lipids, the fatty acid chains are there. Here it is the isoprene chains are there. Okay, and for further differences you can see refer to this chart. I hope this is clear to you. So this is all about the prokaryotes. All right, so LUCA is divided into three domains: bacteria, archaea, eukarya. Today we have discussed these things. These are the prokaryotes. Okay, and eukarya are basically the eukaryotes which contains the protista, plantae, fungi, animalia. Okay, I hope this is clear to all of you, and if you have enjoyed this video, please share this with your friends. All right, and subscribe to my channel. In the next class we are going to talk about the eukaryotic cell organelles and the functions. Okay, thank you for watching this. Take care, have a nice day. Bye bye.